To quantify peptide translocation across vesicle membranes, fluorescently labeled lipid vesicles are generated with encapsulated trypsin. A trypsin inhibitor is added to inhibit the activity of any exogenous trypsin, leaving active trypsin in the vesicle lumen only. When fluorescently labeled peptides are added to the vesicles, their fluorescent tags act as FRET donors and excite the fluorescent molecules on the vesicle. However, when proteins traverse the membrane, they are digested by trypsin, and the FRET signal is lost. Spectrometric analysis reveals the extent of peptide translocation, which is seen as a decrease in the FRET between the vesicles and the peptide over time.
The main advantage of this technique over other methods, including other lipid vesicle techniques and methods to measure peptide entry into cells, is that it gives information about the kinetics of membrane entry, and it does not require the addition of a fluorescent label to the peptides if they contain a tryptophan.
This method can help answer key questions in the design and engineering of membrane-active peptides, such as which characteristics are necessary for effective membrane translocation.
The first step of this procedure is to prepare large unilamellar lipid vesicles, or LUVs, to serve as cell membrane mimics for the assay. Begin by combining phosphatidylcholine, phosphatidylglycerol, and 5-dimethylamino-naphthalene 1-sulfonyl phosphatidylethynolamine in chloroform.